Thank you very much, and hopefully everyone is well caffeinated now. I know I am. So I am going to talk to you folks about the work we're doing at Stanford, which we call Integrative Personal Omics Profiling, or IPOP. And so kind of the general goals of the study fit in with a lot of what's going on here. I would say we study nutrition, but that's not necessarily our primary focus. But so overall, what we're looking at doing with our studies are really deep biomolecular studies over long periods of time in individuals to really get a better understanding of A, what it means to be healthy, and B, what it means to be ill. And so illness can mean very many things, from a common cold or flu to things like diabetes or eventually cancer. And so one of the primary focuses that I'm going to talk about today is obesity, and so how that affects individuals on a more personalized level and a more quantified level. And it was really exciting to see many of the talks at this conference. We're really talking about wellness in a more quantified way, and I think that's where these fields really need to go. And so to that respect, we're trying to do that in a very comprehensive fashion using most modern technologies available in biomedicine. And so what we study what we call the integrome. And so what that means, so I'm a geneticist, so we start, we're DNA focused, so sequencing individuals' genomes, but right, your genome does stuff, and so the transmission of genetic information through RNA to make proteins, and so proteins do your work in your cells. And so there really are some very comprehensive means of measuring each of these things via omic technologies. So things like genomics, transcriptomics, and proteomics. But on top of that, you have all these other levels of regulation. And so we heard a very nice talk right before me about the epigenome, and so how that affects your health in general. Your transcriptome is also modified as well, and so there's this burgeoning new field of epitranscriptomics. That's really, we're just scratching the surface over what these regulators mean. And then downstream, the output of your cellular pathways, so your metabolome. So these are all the metabolites that you produce, but also what are coming from your environment and your diet. And so this particular conference is quite unique in that a lot of the talks have focused on a couple of these things, whereas most researchers today are really study only one of these things in kind of isolation. And so what we want to do is go beyond that, because what we do know is these all cross-talk. So they feed back on each other. And so what we mean to do is generate data on a really comprehensive scale and really gain new biological insights from that. And so it gets really complicated, right? So this is just for one cell, but you can roll this up to the organismal level. And so, for example, we study primarily out of blood. So there's many different cell types in the blood. There's also circulating proteins and metabolites and whatnot in the blood, and so that needs to be quantified as well. And so a lot of the blood is made up of immune cells, so you have your immunome. And so we're an organism, but we're also an ecosystem as well. And so what we also have is our microbiome, and so this is kind of your gut microbiome, but we have microbiome on our skin, in our nose, so all throughout our body. And so each of those different microbiomes most likely feed back as well into this entire system. And so we want to complicate this even further, right? And so we want to measure these things over relatively long periods of time. So taking each of these and then sampling over a many-year period to see how these change in humans and at what scale. And so as far as technology, so the major technologies we utilize in these types of research endeavors are, your standard next-gen sequencing. primarily dominated by Illumina technology these days. So we use that for DNA sequencing, RNA sequencing, epigenome, as well as microbiome. And then the other major technology is liquid chromatography mass spectrometry. So this is a primary driver of things like proteomics and metabolomics. And I'll show some data from those as well. And so I'm a data guy, so one of the main challenges we face in these fields and why these fields are so compartmentalized is that the data looks so completely different. So if you're a geneticist, let's see. Yeah, so you're used to looking at something maybe like on the top here, so shotgun next-gen sequencing reads. And so quantifying and characterizing those. Whereas if you work in the field of proteomics or metabolomics, so this is a trace from a plasma proteome. So this is quite different. So the units, right, you're looking at time off a retention time off a chromatographic column, as well as ion intensity, mass to charge ratios. And so the signal processing is quite different for data like this to data like this. So one of the big challenges we face is how to put these things together. And so, one of the benefits of proteomics, we don't necessarily have this in metabolomics, is you can sequence as well, right? So you have tandem mass spectrometry where you can target specific ions for subsequent fragmentation. So for a polypeptide, you add basically energy, and so that polypeptide fragments at places we know along the amino acid backbone, and creates these kind of characteristic spectra where you can then deduce the amino acid sequence. So like DNA, we can also sequence proteins as well. So metabolomics is much more challenging, in which we can't really infer the chemical structure from what we see in mass spec data. But there are additional ways of using spike-in standards and libraries and things that we can kind of assess the various different biomolecules we're looking at. And so I'd say that we were kind of one of the first groups in the world to do this at a very large scale. And so this started out a long time ago, where we piloted this, basically studying as one particular test subject is the chair of the genetics department at Stanford, Mike Snyder. So Mike was followed for over now 8 years, and so you can then pull out these really dense omic profiles that consist of many different layers, and so including things like genetic variants, changes in gene expression, when you go from a healthy to a sick state, changes in protein. And really this is obsolete at this point because the number of assays we've added onto this is so massive. So how this worked in the kind of initial days is sampling. We'd take blood samples, now microbiome samples as well, and do things like large-scale omics profiling, but also standard medical tests like that you would get at your physician's office. And one of the things we noticed over the course of this multi-year study is that, so Mike got ill a number of times. So he had a HRV infection, as well as a real nasty RSV infection midway through the study. And one of the things that we noticed over the course of that is his fasting blood glucose spiked significantly. And it was really coincident with this RSV infection. And so it spiked to the point, stayed highly elevated for many months, to the point where his physician diagnosed him as a diabetic, which was quite unique. A relatively, a very healthy guy up until that point. And really this proceeded for a very long time, until through intervention via diet and exercise, he was able to bring these values back down. But in terms of days, this is a very long period of time. And so there were hints of that in his genome as well. And then in our multi-omic profiles, you could see changes associated with that progression to type 2 diabetes. So this is just an example of a plot of inflammatory cytokines, chemokines. And you see these spikes when you transition from a healthy to a sick state. And then looking across many different types of omics data, we observe these trends that corresponded with this transition here. And so if you pull out the genes and proteins and whatnot that were associated with this particular trend, and they make biological sense. So for example, this is a trend over time during this period, which was made primarily from gene expression changes, but so was all these particular targets are associated with insulin biosynthesis. Whereas this particular cluster as well, this was a little more different. This spiked up at this particular juncture and then went down. But this was primarily proteomic as well as some gene expression as well. And so this is, again, a similar biological response. And so one of the take-homes from here is, this is primarily gene expression, this is primarily proteomics. So you're getting different information from these different type of omes. So this is, one of the reasons why we want to follow all these different types of biomolecules, because they give you a different picture of what's going on in the body. So where we've gone from there is, so we've taken these approaches and we've rolled this out to, right now, over 100 people recruited through Stanford, where we track these individuals in really great depth over a multi-year period. So initially a 5-year period, but we really like to track these individuals over decades. And so how this looks, so kind of like in the initial pilot study, we sample people regularly. So they come in, give an initial sample, we sequence their genome at this point. And then they come in really a couple times a year and give a set of, so in our case, blood and microbiome samples. And then we want to capture these perturbations. And so, for example, if an individual gets sick, we want to take many samples over that period of time. And so we'll send phlebotomists out to their house and draw blood at that point. Things like stress. So we implement these perceived stress surveys. And so if an individual is undergoing psychological stress, we want to take a large number of samples as well and see if there's changes in the omics data, as well as dietary changes. So again, of particular interest to this group, so when an individual changes their diet, and some of that's experimental, which I'll talk about later, again, taking a large number of samples. And so the good thing about this particular study, so this represents one of the 3 main projects for the current version of the Human Microbiome Project. And the good thing about that, what that means is these data are all going to be made fully public in a relatively short period of time, and that includes genomes as well. So this is, really pretty rare for kind of a large-scale study like that. And so this is, individuals can download these data and run their own informatics pipelines on them. And so we've been at this about 3 years. We're about 3 years into the 5-year study. And so you can see kind of where the patients are in terms of the number of samples that they've been giving. And so, so the earliest patients are now around 30 different individual time points. So you really have a pretty long time scale portrait of how their health changes over time. And so getting into the individual assays, so what do we do when we get these samples? So we do some of the most comprehensive omics profiling that really anyone does. And so I'll go through these one by one. So we primarily sample blood, and so we take blood, separate it into constituent components, plasma, cells, sera, and then run a very large number of omics assays. So from the plasma, we measure proteins both by mass spec and through a variety of targeted means, as well as metabolites, and we do that via mass spec, and that gets you on the order of about 10,000 metabolic features. So from the cells, we extract RNA, DNA, protein. From the RNA, we do RNA-seq, so gene expression. Protein, again, mass spec-based proteomics, and then the DNA. So as I said, when the individuals are recruited to the study, they'll get either their genome sequenced via whole genome sequencing or exome sequencing. And then we do epigenetics on every time point via whole genome bisulfite sequencing. And this is really the most expensive part of the study because you're in essence resequencing everyone's genome at very high coverage at every single time point. So it gets very expensive. And then we look at the immune repertoire using a relatively new technology called mass cytometry, or CyTOF. And then from sera, some of the data I'll show in a little bit, we measure cytokines, chemokines, adipokines, and that's on a Luminex multiplex ELISA-like assay. And so again, I said this is part of the Human Microbiome Project, so microbiome is a huge focus of what we do. And so we also sample stool as well as urine. And so from the stool, we sequence the microbiome different ways. So we started out doing 16S. We're now doing full metagenome. So what is the constituency of the bugs in your gut? And then we go beyond that. And so what do they do? And so that falls under the field of your metatranscriptomics and your metaproteomics. So what are these particular microbes expressing in terms of their genes and their proteins? And that gets you a little closer to function than just having the identity of these things. And so along with that, we also sample a couple different other microbiomes, nasal, buccal, skin. And then along with kind of your standard medical assays, we're doing over 14 different large-scale omics assays. And so we're at the point where we're measuring probably on the order of millions of biomolecules at every single time point for these individuals. So what do we learn? So I'll go into a little bit of a couple of these different types of omes. So first the genome, right? So I'm a geneticist by training. So we start there. So for genomics in our group, so we have an automated pipeline for generating a personal genome. And so we call this HugeSeq. We're at version 2.0. This is fully open source. You can download it and implement it. And what the goal of this is to take the genome, so map the reads, call variants. So this can be single nucleotide variants via GATK, as well as structural variants via a couple different tools, copy number variants. And all that feeds into a number of different annotation tools. So for the changes we see in the genome, a couple different questions. Are they known to be clinically relevant? Are they predicted to be deleterious? Do they have any relation? Are there any pharmacological relationships between the variants we see? And so all this gets output to a report and goes through one of our genetic counselors. And we actually return the data back to these individuals. And that's one of the main reasons the folks like to participate in these studies, is because they do learn a lot more about their health via the genomic sequencing results, as well as the other things. And so, some of the things we've learned just in the initial batch of genomes, so you find relatively rare mutations in folks. So in terms of incidental findings, we find incidental findings. So this is an example of one of the initial 10 genomes where we found an individual that carried a particularly rare SDHB mutation, which this is a healthy patient, but this particular mutation is known to be highly penetrant for paraganglioma. And so at least for the benign kind of head and neck lesions, that we're talking like 80%. And so many of these individuals that does lead to cancerous lesions down the road. So again, so why is that helpful? So these individuals can then potentially undergo additional cancer screenings, and really they can inform their children as well, who can then be tested for whether they inherited that same particular allele. And so of particular interest to this group, so we do dietary perturbations, different types of dietary perturbations. This one was a weight gain perturbation. So this is pretty interesting. So a subset of our individuals were willing to undergo a hypercaloric diet for a period of 30 days. So we recruit them, sample them at baseline. We're talking blood and microbiome. And then we put them on a 30-day hypercaloric diet where they're eating essentially an excess 1,000 kilocalories per day over that 30 days. And so they gain weight. I'm actually always surprised that people are willing to do this for us, but we do have very willing participants. And so after that 30-day period, they maintain peak weight, so getting to a kind of a new homeostasis. So we do that over a week, then we sample again, blood and microbiome. And then with the help of our nutritionist, they're put on a weight loss program to return back to their baseline weight. And then again, we sample blood and microbiome. And so one of the features of this particular study is half the individuals are pre-diabetic. And so all individuals are overweight to start with. And so they're healthy overweight with a BMI of about 25 to 35. And they're BMI matched between the two groups. And so we're trying to not feed them relatively unhealthy things. So how we control this is, so we're not replacing their diet, but we're supplementing their diet with a food preparation from the Stanford Nutritional Kitchen. So we give them a series of snacks, and they're supposed to bring back the empty wrappers every week. And so, like I said, so we give women an additional 750 kilocalories per day, and men get 1,000. And so we have, in terms of the nutritional content of these particular foods, so we're trying to keep them relatively healthy over the period of this time. So we don't have any really long-term ill effects. And so, what do we learn from this type of perturbation? So first off, we can just look at baseline and ask, are there differences across all these ohms between folks classified as pre-diabetic and those that are healthy individuals? And so we do see pretty significant differences, and I'll go into what that is. So this is a picture of a gene co-expression network. So this kind of complicated, but what we do is we take every individual's baseline samples, and we look for genes that essentially co-express, so travel together in expression space, and you can identify kind of these hotspot modules of genes that are highly correlated amongst people. And so then you can go and test for things like association with clinical parameters. And so, for example, a couple of these modules were associated with insulin resistance. So we use a test called SSBG, which is like an oral glucose tolerance test, but a little different. And then we can look at things like the metabolome. And so we see a large number of metabolic features that are different just at baseline between pre-diabetics and healthy controls. And we can do pathway analyses on these as well. And we see a couple different highly significant pathways that differentiate between these two groups. I'll get into a little bit more about what things biologically we find in a little bit. But again, so this is looking at across most of the cohort, looking at cytokines at baseline. So this is a circular plot. So we measure 63 different cytokines, chemokines, adipokines. And so this is just at baseline. And so what you see is really everyone is quite unique. So these profiles are very different from person to person. And then you kind of have these outliers where a particular person will have a specific upregulation of something like, in this case, IP10 or IL-7. And so these people are quite unique. And so we have the added benefit of having multiple baseline samples, right? So we can look at how that changes over time. And so this is plotting the CV of all these things across multiple baselines within subjects. So for multiple samples from a person versus across subjects. And you see that you only see about 10% variation in these things in each individual person. So there's a couple outliers, and these are rather noisy signals, I would say. But across people, you see change. You see differences of about 50 to 60%. In these, and these are just plasma cytokine levels. So we are quite unique, and this is another type of omic data that kind of bears that out. So I said I'd tell you about what we see that changes the most between these individuals. So this is looking at pathway analysis, and the blue lines represent at what direction they're up or down regulated relative to each other. And so for the pre-diabetics, we see changes in genes and proteins that you would expect to see. So, the top hit is a diabetes-related gene. So this is MODY genes. So these are particular transcription factors that are known to be mutagenic in what we call monogenic diabetes. So for most of these folks, we don't observe mutations in these genes, but their gene expression is aberrant. So even though we don't have, even though they don't carry this particular mutation, there's something affecting their gene expression. And so again, so the other things we see, oxidative phosphorylation, a large number of different inflammatory related pathways, immune related pathways, defensins, what are associated with the healthy controls. We see a large number of genes related to olfactory signaling, and so olfactory receptors and whatnot. And this is particularly interesting because this is in the blood, so we're looking at essentially smell, but in blood cells. So kind of interesting. And so, like I said, so folks gain and lose weight over that time. So I apologize. This is plotted in pounds, which is the relatively obscure way we measure weight in the U.S. So this translates to about an average about 3 kilograms over the course of the study. So it's relatively modest. They're not gaining a ton of weight. And then you see most of the individuals return to that baseline weight by the 60-day period. Some of them take a little bit longer. And then we profile that as well. And so I want to talk for a little bit about how we do the integrated analysis. So everything we do, again, starts with the genome. So the genome can inform many different things, aside from just calling variants. So what we do with the genome sequence is we use it to map your proteomic data. And so we generate what's called a personal proteome. And so this basically identifies novel protein isoforms that you wouldn't necessarily see if you mapped proteomic data back to the reference human genome. Same thing goes for transcriptomics. So we map our RNA-seq data all back to a personal transcriptomic reference generated from each individual's genome. And so this doesn't buy you a lot, maybe on the order of a couple % of novel things. But again, this is, those are the things that are potentially the most interesting to follow up on in these big omics studies. And so harder to connect metabolomics back to the genome, but what we do is identify defects in metabolic pathways from the genome sequence and really look for changes in the metabolome or the lipidome via these connections. So does someone that has a mutation in a specific gene have an up or down regulation of particular metabolite associated with that? Then we do a lot of normalization on these data types. So we do, it's very specific to each data type. As I told you, they're very different. So we have these specific normalization pipelines. So how you normalize proteomic data is quite different from RNA-seq metabolomic data and microbiome as well. And then there is a step where we take all the data, scale it, do some additional transformations and weighting, and really get an integrative omic data set that we can then mine together. And so from that we do fairly standard kind of downstream analyses, so machine learning. So do we see patterns in the data that go across different types of omes? We do time series analysis, right? These are longitudinal studies, and really look across different types of biological information, public data sets. We're one of the big data centers for the ENCODE project, so we can relate a lot of the data we see here back to regions in the genome that are well annotated. And then you can identify, you can pull these interesting stories out of these data. So this is looking at metabolic data from the plasma. So your metabolome, so we're tracking these people for 5 years. So we can't expressly tell them that they can't take painkillers if they have a headache. We tell them to avoid them before coming in and giving a fasted blood sample. But then you can go in and look at the data in the metabolomic mass spec data. And so there's different patient samples, and you see spikes for things like acetaminophen and ibuprofen. And so this is particularly, these are the types of things that you'd potentially want to flag. So especially in the case of ibuprofen, if you're studying something like inflammation, for example. And so then what does this look like, when the folks gain and lose weight? So as I said, this is kind of the course of the study, and this is the actual physiological data. And then you can find changes in the omic data that look like that. So this is looking at biomolecules that go up over time, so red going up and then coming back down, and the vice versa, things that start out high go low and then come back. And so we can observe these patterns. We can then extract these patterns from the data via various clustering schemes, and then you can annotate them as well. So for example, this particular cluster is of things that go up and come back down, really kind of mirroring what happens when these folks gain and lose weight. And you can test for functional enrichment of the particular biomolecules in here. And so this particular one is primarily made up of mRNA, but all these factors are related to positively regulating for the innate immune response, which is particularly interesting. And so again, looking at metabolic data, so this is kind of the baseline, but when these folks gain and lose weight, you see changes over time. So phospholipids go down and then come back up, as well as do carnitines, and there are additional signals as well. And so then kind of as an overall look at what changes, so when individuals gain weight, so kind of looking at that same, what goes up and what comes down. So when folks gain weight, even this relatively short-term weight gain, we see things like inflammatory responses. So this again ties back into some of the earlier talks that we saw. So even a modest weight gain induces kind of a systemic inflammatory response, as well as other things. Again, we hit these olfactory signaling pathways as well, so particularly interesting. And so the things that go down when folks lose weight, a lot of factors associated with platelet activation. It's particularly interesting as we also observed this in the original pilot study of Mike Snyder. And platelet activity is known to be regulated somewhat by insulin, so potentially makes sense. And so when folks lose weight, the take home is these things completely reverse. So all the things that go up when they gain weight are back down when they lose weight, so and vice versa. So really, the short-term weight gain may induce things like inflammation and whatnot, but the good news for folks is it's largely reversible. We talk about the microbiome. So again, we see changes in the microbiome over the course of this weight gain perturbation. So this is a little complicated, but I'll say, so each of these different colors on the bottom represents a different individual, and there are different time points over the course of this study. And one of the main take-homes is you can see that everyone's quite different in terms of their microbiome. Your microbiome is very unique to you. Though we do see, in the course of this, we do see changes related to the weight gain perturbation. And so this 16S data at this point, and this will soon replace this with metagenomic. But what you can do is then you can look specifically at, so this is the overall microbiome, but we can look specifically for changes that are associated with our phenotype of interest. And so, for example, looking at IR, pre-diabetics versus healthy controls, we see changes in particular microbes across the entire cohort. So these are in common. And so particular genus within the Firmicutes phylum. So Firmicutes has been known to be associated with things like obesity. These two groups aren't, different. They're BMI matched. And so the only difference between the two groups is subsets pre-diabetic. So these specific genus are associated or particularly upregulated in the pre-diabetics, which is potentially pretty interesting because you could potentially think of making a diagnostic that's microbiome based, for example, for prediabetes. And so where we're trying to get with this is getting towards a more comprehensive modeling scheme for metabolic disease. And so this kind of ties back into the overall theme of this program and a lot of the other talks. And so that spans things like individual variation in gene, in variance, gene expression, as well as different ohms at baseline, for example. And then when you have these perturbations like gaining weight or taking a supplement, what changes? And so in our case, we see things like inflammation in circulating cells, changes to free plasma proteins and metabolites, and as well as a remodeling of the microbiome. And so this is really what we're hoping to get towards, is a more comprehensive view of what it means to be healthy versus what it means to be sick. And so as a conclusion to this, and I'll give a little teaser of some new stuff that we're doing in addition, is as you all know, every individual is biochemically distinct. And our studies kind of really bear that out. And that goes across every type of omic data we generate, as everyone is very, very unique. However, you have these similar responses to these perturbations. So in our case, this hypercaloric diet, potentially taking supplements. So we are in common in some respects. And the challenge is figuring out the differences between people, and so how people will respond similarly to certain things. And again, so even a very modest short-term weight gain is deleterious. So it increases inflammation that we observe, across gene expression, across proteins, across cytokines, and things associated with the microbiome. But again, it seems to be reversible. So that's the relatively good take-home for us. And so moving on to additional studies that are pretty cool. So one of the ways we're implementing this current method is what we call the NASA Twin Study. And so there's a collaboration with a number of other groups in the US where we're taking this multi-omics strategy and studying an individual living aboard the International Space Station. And so we're particularly lucky in that we have a set of astronauts that are monozygotic twins, so their genomes are identical. So the changes that we see are potentially all environment. And so we have one twin living aboard the International Space Station. He just returned about a month ago. And we've been sampling him over that 1-year period, while his brother lived on the ground and went about his life. And we sampled him kind of simultaneously. And so we can pull out interesting things. So again, the International Space Station, one of the really triumphs of collaboration between Russia and the US. So we're shipping samples down from there. So a SpaceX vehicle just recently docked a few days ago up there to bring our samples back down. And so that goes to NASA, JSC in Texas. And then his brother sends samples from home. And so some processing gets done. And so all that data gets shipped, or all those samples get shipped across the US. Different omics profiles are generated, and then all the data is sent to Stanford, where we do integrative analyses. And so the overall take-home is, right, space is a vastly different environment. And we don't have a ton of data back yet, but in terms of kind of what we're looking for is, right, so this is a unique way to study how the body responds to microgravity, higher background radiation, non-24-hour light DART cycle, and many other factors, including nutrition, right? So the food they eat has often been freeze-dried or irradiated, and so they're living in a relatively microbe-absent environment, and the food is relatively free of microbes. And so we can do additional things like study vaccines. So this is an example of, to what I understand, the first administration of the flu vaccine in space. So does the immune system respond to a perturbation differently in outer space? And so like I said, this is, we're only studying one individual at this point, but the eventual goal is to really understand how people respond to living in space. And to get beyond that, we'd really like to study everyone, aboard the International Space Station. And that would really take a real collaborative effort between Roscosmos and NASA. And so hopefully that's something we can do in the near future. And so as kind of thanks for the various folks that do the work. So I'm in Mike Snyder's group at Stanford, and so this is our IPOP team, and we have numerous collaborators that we work with. And again, this is part of the Human Microbiome Project, as well as my funding as well. And so again, like I said, the data, especially for the dietary studies, we make fully kind of open to the public, and that'll be hosted in a couple different places, this particular website here, and as well as the Human Microbiome website as well. And so folks can email me if they want more information. But overall, yeah, thank you for allowing me to speak to you guys.
Thank you so much for this absolutely wonderful talk. It's very, very interesting. Guys, we are a little bit behind the schedule. We have time for one question. Yes?
Да, хорошо. Просто невероятная по масштабу работа, по видимости, ну вот, произведена. Я хотел спросить первое, это по поводу гормонов в крови разных, которые вот там хроматография, как я понимаю, была, но хроматографии-то не все. Какие гормоны анализировались в крови при вот этих состояниях? И хотел спросить, там каждый раз, как я понял, геном секвенировался заново. И в этом контексте, во-первых, идеологически, что ожидалось увидеть вот при повторном секвенировании генома не отдельных клеток, а, как я понимаю, все равно большей части клеток. И известно, что костный мозг, главный продуцент стволовых клеток, он постоянно, ну там наблюдаются мутации с возрастом, они влияют на состояние генома крови. Наблюдалось ли вот это, какие-то такие паттерны в их исследованиях?
Спасибо. I will translate. So first of all, this guy who asked the question expressed the feel of being amazed by all this complexity which was profiled in all those people with various kinds of methods of profiling. And he has two questions. One of them specifically on the hormones, which you probably profiled in blood, and what was the method for looking for hormones in the blood specifically, because there are different group of hormones present there. And second question was on repeated sequencing of the genome, because there are possibilities of mutations within the blood marrow cells, and they would give different clones in the blood. How this part was assessed?
Oh, yeah, so they're both very good questions. So in terms of the first question, so hormones, so that data aren't fully ready yet at this point. So a lot of hormones are peptides and whatnot. So some are metabolites, so we can measure some of them via metabolomics, and some of them we have to do kind of a peptidome measurement, which we haven't completed yet. So I can't really give you the full story over how hormones change in the body. But as far as your second question, so yeah, that's a huge challenge. Somatic mutation. So looking for somatic mutations in blood cells. We've thought about doing it, and so one of the things we're potentially, so it's tough to do. We're applying single-cell sequencing approaches to potentially. Yeah, so two approaches. So single-cell sequencing, which again is not getting you a full genome per cell, so very challenging. But also taking blood cells from these people, building iPS cells, which you can then characterize as well as people change over time. What the goal is, of course, over a many-year period, a lot of these pre-diabetics are going to convert to full type 2 diabetes. And so how do their cells change over time? And so that can be mutations, and that can be many other things.